Well, first I'd like to thank Professor Zhao for that wonderful introduction to the session and for introducing me, and also to the organizers for giving me this opportunity to share with you some of the work that we're doing at my laboratory and at Georgia Tech in general. And I've shortened the talk title to just exploring alternative routes to the first RNA-like molecule, RNA-like polymers, because it's a little easier to read, for one thing. And the other is that I've broadened the topic a little bit with respect to some of the work that I'll share with you. First thing that I'd like to do as part of my talk is to just introduce to you a couple of centers that we have at Georgia Tech, and I hope that you've had an opportunity to see some of the posters by colleagues from the center that either at Georgia Tech or also at other institutions. And I don't have time to go through all of this work yet. I'm involved with this in the co-advising of students and postdoctoral researchers. The first one that I'd like to mention is the center that we have called RiboEvo, that is focused on understanding the evolution and of the ribosome and of the development of peptide synthesis, that is coded peptide synthesis in life. This is a node of the NASA Astrobiology Institute. It's headed by my colleague and good friend, Lauren Williams, right there. We have two posters here. The first author is Jessica Bowman on one, looking at domain structure of the ribosomal large subunit, the search for co-opted RNA structures in the ribosome. What we're finding there is that the folds in the ribosome look to be very modular, at least for some of them, and quite robust. And that actually is giving us a lot of encouragement that we may be able to track the evolution of the RNAs that make up the ribosome, which of course is a very large molecule. Dana Schneider here also has a poster on resurrection of the prebiotic assemblies, that is interactions of ancestral RNA, ribosomal RNA with ancestral peptides. Based upon some modeling that's been going on at Georgia Tech, we think it's possible that we could reconstruct not only parts of the ribosome, but the original peptides that were interacting very deep into the ribosome. And if you see her poster, it's quite exciting. We have data showing that there is actually interactions between some of these modeled peptides and this very rudimentary model of the ribosome, cut down to about 20% of the size of the full ribosome. The other center that we're quite fortunate to have that has been mentioned a couple of times already here at this meeting, the Center for Chemical Evolution. You probably recognize some people there. This is actually a little bit of a dated photo. We've got about twice as many members now. We're a Phase 2 Center for Chemical Innovation. We're quite happy that we are supported not only by the NSF, which supports all of these centers for chemical innovation, but NASA has also provided some of the funding through the exobiology program. This center has a much broader topic than the previous one I spoke about with the ribosome. We are focused on understanding the origin of functional biopolymers, which of course includes peptides and nucleic acids. But we're trying to take some different looks at how this could come about. And a couple of posters show some of our thinking, again, trying to think a little more broadly about the problem. Irina Mamajanov has a poster about nucleic acids in non-aqueous environments. You may recall earlier in these sessions, there's been discussion about the water problem. If you want to make peptides, for example, you need to get rid of water. If you want to polymerize nucleic acids, you need to get rid of water. So one thing that we're exploring is the possibility that we could have had life start in a non-aqueous environment. And for somebody that works on nucleic acids, it's almost heresy to think about a folded nucleic acid in a non-aqueous environment. But what we have found is now that more than one solvent that is water-free can support folded nucleic acid structures. So we think it's very possible that at least some part, say, of a daily cycle, nucleic acids and peptides could have existed in a water-free environment that would have promoted their polymerization. Another poster by Sarah Walker and Martha Grover, myself, is about the emergence of population structure and functional evolution in simulations of environmentally driven replication. There we're trying to see the factors of polymerization rates versus degradation rates in limited diffusion systems. And from there we see that you can actually get a system going even if you have polymers that are only marginally stable, that is just stable enough to get a couple of progeny. So again, looking at a broader view there. So with that, I'll get on to some of the work that we're doing directly in my laboratory. And again, all is quite collaborative work. We're looking at the big question of how do we get to RNA? And I'd say that it's fair to say that the RNA world hypothesis still faces multiple chemical challenges. When we think about the problem, and it's been brought up already at this session, that the prebiotic chemical inventory, as we call it, was probably a very complex mixture of molecules. And we think it's fair to say that it's still unresolved how molecular selection took a subset of these molecules here to somehow partition, selectively interact the molecules that are needed for RNA, and then to correctly couple them into RNA. And I put correct in quotes here because when we look at RNA, we say, oh, this is correct. But as has already been discussed here, there's a possibility that there were different linkages early on. So I don't know exactly what correct is with respect to the earliest RNA-like polymers. So I'm going to broaden that hypothesis out even further, and this is what I like to call a general hypothesis for nucleic acid evolution. It's not just ours. There's a number of other groups that I think fall along with this logic as well. We think that RNA is a highly evolved molecule, and that if we look at RNA, we can think about it as being kind of the cooperation of three different types of molecules. We, of course, have a base, a nucleobase informational unit. We have a sugar moiety, and then we have a linkage molecule or connector molecule there. So when we look at nucleic acids now, we think about these as three different distinct parts that could have come together at different points, and that their evolution did not necessarily have to be coupled in time either. And so if we think of it very simplistically, we could have started out with a proto-RNA that had different bases, different sugars, different connector molecules that were assembled with the aid of prebiotic cofactors that I'll talk with you about in a minute. But this would have been an easy-to-assemble molecule, a very robust polymer scaffold is what we're thinking of here. And then we can think about multiple stages of evolution, such as sugar evolution. We know that from work, say, from Ram Krishnamurthy, Esha Moser, Peter Herdewin, and others, that we can change out that sugar from a ribose to hexoses, for example. So we can change that out, and so maybe life started with a different sugar. We could have an evolution change that sugar. We could have base evolution take place there, and connector molecule. We've done some work with glyoxylate linkages, which of course are a carbon analog of phosphate, so we think that that could have been replaced over time. And when we look at this molecule here, we see this as functionally superior. It's amazing what RNA can do. All you have to do, again, is look at the ribosome and see the machinery that can be built out of RNA. And so when we look at a molecule that's very difficult to synthesize but works very well, that to us looks like the hand of evolution at work. And of course, this is now made with the aid of proteins and enzymes, protein enzymes. Now, for those that might question whether or not RNA evolved and say that doesn't seem likely, what I like to point to is that DNA, right, is probably an evolution of RNA, right? We have RNA losing an OH, and so that's a change in the sugar to become DNA. That's becoming functionally superior for what it does, long-term storage of information. And we also have the change in a base taking place, going from RNA to DNA. Again, kind of a mechanism, it seems, for protecting information. So in our mind, going from RNA to DNA is essentially the same as going from, say, some earlier proto-RNA to RNA today. And I just have a citation here. There's I think a very nice contribution. David Deemer, Jack Shostak edited a book with Cold Spring Harbor, Origins of Cellular Life, where Aaron Engelhardt and I wrote a chapter where we tried to summarize different parts of this type of evolution, bringing information from different laboratories. Okay, So we also follow in our laboratory some questions, paradoxes. We try to solve these, reconcile them with the chemistry that we think is feasible. And one of these is what we like to call the paradox of base pairing. And this is just one of several challenges that I see to nucleic acid evolution. And that is this: if you have oligonucleotides that are of complementary sequence, you know that they will pair and form duplexes. There's no problem there. The problem comes in is if you look at monomers, mononucleotides or the single bases, they don't form base pairs in aqueous solution. And that's a problem. It's a problem for me with respect to evolution, because evolution, as we know it, doesn't predict what's going to be needed in the future, right? What evolution does, it takes something that exists and it starts to use it for different functions, right, and makes small changes to it. So I have a problem with the idea that polymers were spontaneously forming with the nucleobases before they were base pairing, because I think that we need a selection mechanism to get just those bases that are going to make these type of base pairs. And if we're going to do that, we're going to do that at either the monomer or at the very short oligonucleotide level. So that's what I see as a paradox. These solutions that we came up with quite a while ago that we're working on constantly in my lab is the idea that intercalators could have played a role in selecting base pairs and stabilizing base pairs. So I'd say here that intercalation is a classic example of molecular recognition. It's really one of the earlier characterized interactions or molecular recognition of nucleic acids. Dates back to the 1960s. And not only is it a mechanism by which small molecules recognize that this is actually a duplex of nucleic acid by the stacking interactions between the small molecule and the base pair, this is also a mechanism by which the base pairs become stabilized. There's a free energy associated with this binding, and so that makes these base pairs more stable, the duplex is more stable. So to us, that's a very simple principle that may have existed all the way back to the original selection of the base pairs. This is actually a crystal structure that we solved recently at Georgia Tech. Kind of happy about that because this was one of the first intercalators that was demonstrated to bind DNA. But the crystal structure had never been solved, and for certain reasons we wanted to know exactly what this structure was. So with help from Lauren Williams, we got the structure recently. So here's the big picture of what we call this molecular midwife hypothesis. Here in blue represents intercalators, and in red and green these are base pairs. And our idea is that these intercalators will help form these stacks. Now these stacks could have oligonucleotides part of them or free bases as part of them, bring together these columnar stacks, again with the molecular recognition between these intercalators and the bases. And then some chemistry, perhaps during a dry part of the day or with some activated molecules around, we would form the backbone. And then what we'd have is an intercalated duplex, and then when water activity is higher, because these molecules, these intercalators, are non-covalently interacting with the base pairs, they can be released out in solution. And when they are released, then the backbone is actually twice as long as it needs to be. And that makes the helix coil up like that. Because if you look at the length of the backbone, even today in RNA, between the connection point of the bases along the backbone, it's actually twice as long as it needs to be. It's 6.8 angstroms there, which is exactly what you have in an intercalated complex. So rather than just philosophize on this, we are looking to see, can this chemistry really work in the lab? And so one of the problems that we've attacked with this is the problem of strand cyclization. Again, it's nice that actually this has been mentioned already at this conference. John Sutherland mentioned the problem of making di and tri-oligonucleotides and having them cyclize and trying to block that cyclization. So we've been working on this problem well. Cyclic oligonucleotides, so those are the cyclic here hexamers coming together. And so that's not what you want. Here's another one from the Ferris laboratory. They've had great success, and I think you'll hear more of this today about using clays to polymerize nucleic acids. You can see that though this is not immune to it, they're still getting some of the dimer species and trimer species where the relative yield of the cyclic product here is substantial. So that's still a loss. And this is a loss even when you're employing a macroscopic surface, which doesn't have such selectivity for the bases as a means to suppress cyclization. So what we did was we wanted to see if we put in an intercalator, rather than go to these cyclic structures, could we make these polymers here instead, being assembled by the intercalators. So we put together a chemical system, a model system. We actually worked here with DNA. It just made things a lot easier. And this is the oligonucleotide 1, as we call it, ON1. And this is, with the sequence CGTA, this does this sliding complement, so we can get, with one oligo, we can make polymers. Idea being is that if these assemble, then our phosphate and our OH at the other end of the molecule, excuse me, will be in close proximity. And let me get a drink of water. So these, excuse me. These will be in close proximity here. The intercalator will bring, excuse me, wow. All right, so these will come closely together here. The intercalator is stabilizing this duplex here, and this will be where this ligation will take place. Now we're going to use some coupling chemistry that's not prebiotic necessarily, this cyanamidazole activation here, just as kind of a reporter. So we're also going to use an intercalator that's not necessarily prebiotic. This is ethidium. It's just very well behaved, very well characterized for binding nucleic acids. We'll put those in with our nucleic acids. And what you see is right away a big difference. This is our cyclic former production in the absence of an intercalator. That's very efficient. This is cyclic 8-mer here, and what you see is that the linear 8-mer is almost not detectable, which tells you that if you do have any coupling at the linear form, it cyclizes very efficiently to cyclic 8-mer. In stark contrast, if the intercalator is present, what you see is you're building up a linear 8-mer, and you basically shut down the production of the cyclic former and the cyclic 8-mer. Now those are the short pieces. If we put this on a gel, we let it run a little longer so we can see the bigger pieces here. What you see is that in the absence of an intercalator, 0 here, you don't see any production of what are these 16 mers here. Even down here in the 8 mers, you're not seeing anything. In the presence of the intercalator, what you see is that you get polymers here that are actually in excess of 100 mers. So this is working very efficiently. It also respects Watson-Crick base pairing. If we have 2 oligonucleotides that can't form Watson-Crick base pairs unless both oligonucleotides are present, here it's CCTA and GGTA, then what we find is that if we have just one of those oligonucleotides in the presence or absence of the intercalator, no polymers. If we have the other one in the presence or absence of it, no polymers, maybe some background reaction of a different type, but not the polymers. But then when we have it in the presence of the intercalator, we get polymers. Again, without the intercalator, we don't get polymers. So it's actually very efficient, and again, respecting Watson-Crick base pairing. So this is not just some random polymerization going on. But I think it's also important, and I think that I have to credit the reviewers of our manuscript for this, for pushing us on this. They said, well, how powerful is this? Can you quantitate that? So we really pushed this at the limit. We had put in there that we can get it in excess of 100-fold improvement, maybe 1,000-fold, but they really wanted a number. And so what we found is that we actually get better than a 10,000-fold enhancement of the polymerization. The reason we have that number, most of our work that we're showing you was done at 0.2 millimolar or equivalently to 100 micromolar shown here. I'm emphasizing that the units are different on this gel right here. But what you see here is that we can take this oligo here up to 60 millimolar, and we're not getting polymers. Yet we can take it down to 5 micromolar in the presence of the intercalator, and the polymers are there. They're working just great. So we get a 10,000-fold at least boost in polymerization efficiency here. Okay, so what we also want to do is test whether we can use one of these intercalators to promote a different type of base pairing, to see what we call an orthogonal unit, where a base pair that promotes Watson-Crick will not promote a different type, and vice versa. To do this, we made use of an AA pair that we had discovered a few years ago to be stabilized by a molecule coralin, and another one called AZA3 in a series of papers. And what we did again was we took oligos with runs of A's, and what I'm showing you here is that if you put ethidium in there, you don't get polymers, maybe one coupling, very small amount, and proflavin maybe one. But when coralin is there and AZA3 is there, you can see you again get very efficient polymerization, showing that molecular recognition. As I said, this is orthogonal because you can take those two molecules that we had before, coralin and AZA3, and they don't, excuse me, they don't promote Watson-Crick base pairing, and they don't promote polymerization. So I just then like to go and bring up the idea about whether the bases were those that we have in life today and whether a transition took place. And one hypothesis that we're really interested in is whether it was a purine world or a pyrimidine world. Was it really a mixed Watson-Crick to start with? And so if we just go and look at the purine base and say that we can have substitutions at these 3 positions and just say that they can be hydrogen, NH2, or oxygen, you know, this right here then leads to 27 possibilities. And this again comes back to the question of, if you're producing all of these in a prebiotic soup, how did you sort out those that are making good pairs to start with? So I'm focusing here on 4 of them. You can recognize adenine, guanine, hypoxanthine, and another one here, which is isoguanine. And we find these ones quite interesting because these can mimic Watson-Crick base pairs. And this goes back, this is a proposal by actually Francis Crick himself back in 1968, where he said life might have started with purine-purine pairs because they had seen these in wobble base pairs between tRNA and messenger RNA. So we like this idea. And Battersby laboratory and Switzer's laboratory in more recent years have shown that you can make stable duplexes with these types of base pairs. So we wanted to see whether we can get molecular recognition with small molecules as a first step towards this. Part of this, what we did was we built models using the amber force field, and it's really hard to tell these two apart. This is a Watson-Crick structure here. This is a purine-purine structure here. There's no high-resolution crystal structure of these yet. We've got crystals. We're going to try to get a structure of it. But this model right here, you can see that the helical pitch and everything is very, very similar for a purine-purine base pair. And so we tested a number of small molecules. This is just a subset of those that we tested for their binding to a purine-purine duplex. And what we find is that we can get a substantial stabilization of these duplexes. The duplex itself is actually less stable than a Watson-Crick duplex. This is actually a 12 mer here, and it melts a little bit above room temperature there. But with molecules, particularly one ellipticine and coralin, we can shift that melting temperature up above about 40-some degrees. So we can shift it more than 20 degrees C with these small molecules. And so we think that this is actually potentially optimal with respect to origin of life scenario, because we may only have this base pairing information transfer when these small molecules are at high concentration, and then when dilution sets in, say when water activity goes high and these small molecules dissociate, we could then actually have strand dissociation folding up into, you know, proto-ribozymes and that. So this might be a way to get around also the strand inhibition problem by having base pairing that's not nearly as stable as it is in the presence of a small molecule. So I'm going to wrap this up real quick here and say that I pushed you into the direction of saying maybe it's just a purine-purine pair to start. I also want to let you know that I really try not to get dogmatic, and we should always be careful if we get too enamored with our own ideas on it. And so that's why I also thought we should back up and look at pyrimidines as well. And I think it would be absolutely premature to dismiss the possibility of a pyrimidine base pairing scenario. And I point out here, pyrimidines are even richer in their diversity because there's 4 positions that you could substitute on a pyrimidine. I'm sorry, I could hardly fit them on the slide, because if you substitute at those 4 positions with just hydrogen, NH2, and oxygen, of course, you end up with 81 different possibilities. Again, this could be a real challenge for the prebiotic chemistry to select out these bases. But there's some interesting ones in here I want to point out. This one right here, 2-pyrimidinone. These are sitting right next to our the 2 Watson-Crick pairing ones. This is the C and the U that we know and love. And then down from these 2 are substitutions at this 5 position with the amino groups there that are very interesting for another reason. This other reason is that these have already been shown by Krishnamurthy and Eschemoser and colleagues to form actually pretty stable base pairs. These 12 mer duplexes are stable 15 to 38 degrees C if they're connected on a peptide backbone. And I think that that's quite interesting, because it seems that if you tried to make these pairs, as far as I know, with your regular ribose phosphate backbone, they're not going to be as stable. But if you change the backbone, you can get a stable base pairing here, which I think is interesting. So that's quite promising. Something else that I like to bring up that's from our laboratory a few years back now, we found that this molecule, 2-pyrimidinone, that if you add that to ribose and you heat it up, you make nucleosides in good yields. In fact, Heather Bean found that she could make up to 12% of the beta-furanose of this form. And so this makes me think that the nucleoside problem is probably not as big of a problem as we've all thought it was. If we change the base, if we change the sugar, if we're not so fixated on it being the canonical base and the canonical sugar, then I think that this coupling reaction is not such a problem. I was going to talk to you a little bit about reversible backbone linkages. I'm going to have to really jump over that. This is in a collaboration with David Lin and his laboratory at Emory University. I won't go through the chemistry that we're using here. Let's see. What I will just tell you quickly is that it's addressing the strand cyclization problem again. We have some chemistry that allows us to couple together oligomers into polymers. In the absence of a template and an intercalator, these will cyclize, the same way that I was talking about small oligos doing that. And now what we're showing is that if we add intercalators and template, that we can assemble these into polymers, and we get that reversibility. That's actually shown here in a gel. And again, just very quickly, if we have this bifunctional oligo right here, and we have it in the absence of a template there, we actually can get the generation of this cyclic product. But if we put in the template with the intercalator, again, we get polymers. So we're essentially able to kind of recycle this cyclic product there by adding in the energy of the binding of the intercalators. Okay, so the conclusions are from the work that I presented here is that I think it's fair to say that intercalation-mediated assembly largely solves the problem of strand cyclization. It's a possible solution. And that intercalators might have selected the original base pairs, either Watson-Crick pairs or their predecessors. I don't want to get too hung up on them having to be Watson-Crick to start with. And then again, briefly, reversible backbone linkages can also help circumvent that strand cyclization problem. These are the people that did the work that I just discussed here. Aaron Engelhardt is a former graduate student that is now in Jack Shostak's lab as of about 2 weeks ago. Eric Horowitz is up at UNC as a postdoc. Irina, I mentioned her earlier here. She's doing this DNA and RNA in non-aqueous environment. Reagan is leading up the purine work. Denise Aniqua modeled the purine structures, and this was done in collaboration with Lauren Williams, as I mentioned. Briefly touched on the fact of what Martha Grover's doing with modeling, that she has a poster. Again, the backbone chemistry was done with David Lin and also with Ram Krishnamurthy now. Others that have contributed are Michael Chin, Michael Smith, and Casey Quarles, who are actually great undergraduates, and Sarah Walker, who is actually an NII fellow, has been doing computer simulations. And of course, all important, we have funding from NASA, CCI Institute, and Exobiology, and the NASA NAI. And I thank you for your attention.